biostatistics in Sanofi Bachin. She has more than 30 years of experience in pharmaceutical industry. She has worked in Merck and Sanofi. This is Sanofi Bachin is a pharmaceutical industry. She got her bachelor's degree in Shandong University and the PhD in Rogers University in the USA. And she has got several prizes for industrial innovation. And at present time she has collaborated with several institutions and has served as corporate management and adjunct faculty in the statistical courses in Rogers University. So, Professor Zhang, thank you very much for being able to speak in our seminar.
Thank you very much for the introduction. And so I just want to give a little bit background of my company. Sanofi is one of the largest global pharmaceutical company, and with the headquarters in Paris, France, and then North America headquarters in Morristown, New Jersey. And the company now is focusing on the immune diseases. So we have a blockbuster Dupixent with multiple indications. We also develop a vaccine product. So I worked in the pharma in Sanofi for 15 years, and then for past 20 years I worked in vaccine. And then I worked at Merck while I was pursuing my PhD, and then worked there 10 years. So my experience across from early discovery all the way to late clinical study submission. So now I'm focusing on the translational biostatistics. Translational meaning you focusing on learning the disease, how it's caused. So sometimes it's by the mutation of the gene, sometimes some protein mutation. So and then from there you will screen all of the compound you have in your library against the particular disease target. Once you find the potential compound, so you will test its efficacy, safety, first in cell level. Cell level is in the tube in the lab. And then you move to animals. And then if animals showing both safety and efficacy satisfactory, then you move to human clinical trials. So in human clinical trials, you go through all the stages. The first stage is to show the dose response curve. So as you increase the dose level, you see the PK/PD. PK/PD is more like in your blood, you see the druggable components. It's increased as your dose level increase. And then once you see it can reach to your body, and it can also need to be able to eliminate from your body. If you cannot have the drug eliminated from body, then it will be accumulated in your liver, then cause the disease. So development of a new treatment is a very long process. And anywhere wrong, and then you would not get the FDA or EMA. EMA is European medical agency. FDA is the US agency. So it's a very regulated process. And then data scientists and also statisticians, the role is to design the experiment and to summarize the data and then prepare the package. So whenever you have a question from FDA or EMA or other agent, you're able to defend yourself. So there's an increased need of us as a quantitative scientist, especially when the big data become an issue. So now even like when we recruit a patient, we use AI-based methodology to see around the region, and can be global site, which season, which clinical center you're most likely to recruit a patient. So using that, you can have a fast enrollment. And also in any other, like you may have some measurement in the past. It's always been patient going to the doctor's office and measure on site, but now they can have the wearable devices. And then the device can be uploaded to the server. Then from the server, go to the central database. Then we could look at the data. So as you can see, data scientist has increased the impact of drug development. So here I'm going to show some steps of new drug development and how data scientists and the statisticians can play a role. And what are the methodology in the scientific methodology, and what's the data scientist method to summarize data, and the challenges and the approach. So if you have a question, you can stop me, ask me a question, and then I will answer it. And so here I will mostly focus on the drug discovery. So discovery is like from the very beginning. The development is more standardized. Development meaning you already find the potential treatment, then you test in human. And then you already know which measurement to collect. You just need to pack them. But in the early stage, many unknown questions. And so this is the whole process of a new treatment development pipeline. So the first one is the target identification and validation. And the second is target identification, meaning suppose somebody got a disease. So what's wrong in the person's body? In general population, what caused that? So that's the fundamental question. Once we know the cause of the disease, then we are able to find something like you have a lock, then you have a key. So learning what caused the disease is the lock. Then we find either chemical compound or biological product or gene therapy. That's your key to match with the lock. Then you could get the treatment, right? So the first is find the disease cause. The second is find the treatment. That's we call lead identification. And then we also need to, the third step is suppose you find some chemical candidate which is potentially can be used as the treatment. However, you need to optimize it with many aspects. The optimization can be improve the safety performance, or improve the efficacy, or improve the stability. For instance, if you go to the doctor, it's in a medicine, you look at the bottom. In the bottom it said storage time, expiration date, etc. So you wanted to find a treatment which can be stored in regular room temperature for certain time. If it's very, like for COVID vaccine, initially you need to store in the refrigerator with very low temperature. So that's very challenging to transport and to the normal pharmacy, right? So what you want to do is optimize it. Let the treatment being easy to store, easy to transport, still maintain the desired efficacy. So that's called lead optimization. And then what you need to do is the preclinical. Preclinical meaning, because of safety concern, you cannot directly test the new treatment in human. It's not allowed. So you have to test it in animal. So animal you could have any animal models, but you need to prove 2 of them showing the desired safety efficacy. So the easiest to obtain is the mouse. Okay, the mouse, you can engineer the mouse with a certain gene deficiency. Then you want to test whether with this gene deficiency the disease occur. And then sometimes you on purposely knock out some gene, because in the first step you identify the gene which mutated caused the disease, right? To prove it in the animal model, you will engineer the animal with some disease gene deficiency, then to test. So we call it wild animal, meaning the original animal, then knock out animal with that gene deficiency to prove your theory is good. And then after that, you can also use the animal model for the safety testing. And then you increase the dose level, and you see whether you observe, you observe a lot of, so what we test in the safety, many things you need to test. For instance, reproducibility, meaning like if young women take it and would not impact the pregnancy. And then also the cardiovascular, the heart disease, you need to test. Or also the liver disease, you need to test. And then you also need to test the cardiovascular disease. So that will take 5 years, because once you take the medicine, the disease is built very rare situation, like maybe 1,000 patients, only one case. So you need to have very large studies to observe certain number of events in order to prove it's safe or not. Okay. And then from preclinical to phase 1, phase 1 is first in human studies. So phase 1, what you do is you need to find a dose level. So you know the treatment is working, but at what dose level? Like each day you take 3 mg or 5 mg, right? The quantity. So that's through the phase 1 study. It's often being used in the healthy volunteer. So a lot of time using the medical school student, because you conduct the clinical trial mostly in medical school, and then you have the availability of the student, they volunteer, and they sign the agreement, and then consent form, then you test in human, healthy human, to observe a safe profile. And then from there you find the dose level, then you move to the second phase. And so over there you will have a dose escalate study. Dose escalate meaning you have a safe dose identified in phase 1. Now you need to test it into the disease patient. And disease patient you need to cover a wide range of populations. So for instance, different race, different age group, gender, etc. And then for elderly, you need to be very careful. So you first give 5 patients, you observe the safety, no issue, then you have the main cohort. So phase 2, you also is a very important stage testing in real patient population. And once phase 2 successful, then what you do is you will have the best is a single outcome, meaning you just measure one medical outcome, which will indicate the efficacy. In phase 1, phase 2, or preclinical, you may have multiple readout. But after all those, you need to find a single readout, at most maybe co-primary endpoint. Then you do a very large population. So how large is the population, the sample size, depending on the medical information. So for instance, you see, let's say we do like a cardiovascular disease. We see if we decrease the blood pressure by 10%, which is an efficacy indication, then with the variability, because each patient behaves differently, you recruit 1,000 patients with their variability, how large the sample size can indicate you have a meaningful medical outcome. That's the first question you need to ask. Secondly, when you do this trial, you have 2 types of errors. One is the treatment is not efficacy, is no use, but you claim it's useful. So that we call the type 1 error. So FDA has a requirement to control this type of error, less than 5%. So if you learn some statistics, you know you use a p-value. P-value often say 0.05, less than 0.05. That's because you wanted the type 1 error less than 0.05. And then there's another type of error, which is called type 2, meaning it's useful, but you say it's not useful. So for that, FDA doesn't care, because it's only the company's loss. It's a good treatment, but you say it's not useful. So it will not be put on the market, but it's not harm the general population. So for that, FDA allowed 20% of error rate, meaning it could be useful treatment, but not being able to put on market. So with those 2, then you will find the right sample size then to do the test. So that's once you show in your phase 3, typically you need 2 studies, both showing positive, then FDA will approve it. So there's a recent news, there's one compound, one side positive, one side neutral, and that one is pending, it's not approved. So you need to have another study to make it be provable. So this is, once phase 3 is launched, and then the treatment will be on the market, but it's not done yet. So there's a phase 4 study, meaning the treatment on the market, you collect the real patient data to see whether it really safe on the general population, even after years of use. So there's a, they call it the post-market follow-up. So this is the whole process, okay? So now I'm going to start on a little bit like the first steps, like target identification, lead identification, and some special user cases to show why big data is occurring in drug discovery and how we can contribute. So any question on the whole process overview? Okay, thank you. Okay, so, okay, I don't want to block, so it's okay, I stand here. Yeah, so target identification and validation is good. And so what's the objective of target identification? So as I said, you identify the genes or proteins or other biomarkers involved in the specific disease process. And so to develop a therapeutical intervention as a treatment. Okay. And the biomarker, so biomarker become very important in drug discovery. So the definition is a characteristic, normally it's a biological characteristic, that is objectively measured and evaluated as an indicator of a normal biological process and a pathogenetic process or pharmacological response to therapeutic intervention. Okay. So it's a very more biologically related. So if you're doing bioengineering or something, this will become very natural to you. So this is the definition established in 2001. And the example of a biomarker, for instance, cholesterol level for heart disease. Okay, so cholesterol level, if it's high, then its indication is the risk factor of heart disease. If you don't control your cholesterol level, eventually you will develop high chance of developing heart disease. So that's why, because cholesterol level you can really measure, but the heart disease may take years to develop. So in drug development, we use cholesterol level. If it's reduced cholesterol levels, certain treatment, then we see it's an effective treatment. And then the other one is HbA1c for type 2 diabetes. Okay. So it's more of the biological biomarker you can measure using certain devices. It's just like, if someone possibly exposed to COVID, then you go to CVS, go to Walgreens, then you have some nasal swab and then test it. So they are testing certain biomarkers over there. So this is the same idea. And then how to find a biomarker. For instance, we know there's a certain disease. So typically what we do is we call the observational studies. So we have a group of healthy subjects and a group of disease subjects. We measure their gene expression and then compare the two different types of population to see which genes being increased or decreased. Then those could be potential target. Yeah, so my query is a technology to measure gene expression. And then later on there are more like RNA sequence, bulk RNA sequence, or single cell RNA sequence, next generation gene sequence. So more and more, there's more technology available. Then you collect even a higher volume of readout with the advancement of the biological devices. And so also it is like a human, every single human, you have more than 40,000 genes, okay, from different tissue, for different functions. And for certain disease, only very small number of genes being up or down regulated. Majority of them not changed. So this concept is very important when you do your data pre-process and analysis. So later on, I will use this concept again. And so what you see, if most genes not a difference between disease and healthy population, so then what you need to do is find those very few genes up or down regulated. So that's what we call the find the needle in the hill. Hill is like the dry grass. So if you drop the needle in the dry grass, it's very hard to find, right? So this is a challenging problem. And then, like if we use a microarray or RNA sequence, we screen like 40,000 genes. We find maybe a small number, but if you have a high throughput, meaning you can screen so many genes, then the sensitivity may not be that good. So what we do is we use a different technology which has low throughput, meaning you can screen less number of genes, but with more sensitivity. So the technology can be different. For instance, we use RT-PCR, ELISA, and or we call the flow cytometry effects. And those are used as a follow-up biological assay method. And then the statistical associated with those can be different. Yeah. So I have a remark is, so for this, as I mentioned, you study a healthy subject versus disease subject. At this stage, no treatment involved. But you do want to have include the disease in different stages. Some is mild, some is severe, and then you also observe when the disease stage changes, like from mild to severe, how the biomarkers you identified, whether it's changed with the disease status. That is another validation that you see these targets are really involved in the disease progression. So even you're data scientist, you're a statistician, if you work for pharmaceutical company, you need to develop a certain biological knowledge. So I worked in pharmaceutical for 30 years, so I have spent a lot of time learning the science itself. Like when I switch from pharma to vaccine, I need to learn how the vaccine development is different. And then if in vaccine, you have to measure the antibody. After taking the vaccination, the people develop the antibody. How the antibody level associated with the disease protection, we call it correlate of protection. So all of those are new knowledge once you switch from one area to the other. Or even like you work in the rare disease, you switch to oncology, then there are many new things you need to study. So my organization supports all the therapeutical areas, focus on the translation, meaning the genomic, genetic readout, how it can serve as the early indication before the real medical indication. Okay, so what is the general challenge for data scientists? As you can see, if you have 40,000 genes, you may only study 100 patients, right? So you have we call large P and small n. So if you have so many variables, very small number of subjects, then you have the chance of the bias. And then you have the chance of overfitting the data, right? So I think in data scientists, you do have the concept of overfitting. And so how do we handle this type of thing? That's a typical issue for data scientists. And so here is a real example of the large P, small n example. So for, when you study the potential target, you don't know it's coming from what. So you study the whole, like a sequence of every possible cause of the disease. So the first is the patient's demographics, like, the age, like some disease associated with the age, like dementia, for instance, right? And then the gender, and we know some is more likely happen to male than female. And then the BMI. So for instance, the body weight could be a factor of the heart disease. And then the smoking status, like COPD, for instance, right? So in general, the patient's demographics always being selected. And then second is the genotype data, which typically is like around 100. And then is the one is common, is genotype, common is gene expression. You can see it's all like already 12,000. So this is the, sorry, this is the fourth rule you can see. So 156, the first number is the 156 patient. Then the second number is the dimension corresponding to that measurement. So if you go all the way down, so you can see you add all together, it's totally, so how to, oh, it's yours? Okay, how to get this one? Okay, yeah, so here, so this is all different type of readout. Then here, the first is the number of subject patient. Then this is the number of dimension corresponding to this type of measurement. So add the second number together. This is how many variables. Okay, so you can see this is really a huge challenge of a large T and a small n. And I just want to know, like in engineering, do you have this type of issue? Yeah, okay. And so then the challenge with the large P, small n, so bringing the computational complexity because data is really large, right? And also overfitting issue. And then the high false discovery rate. For instance, by chance we have 5% of making error. If you do it twice, then you have, thank you so much, you have 10% of making error, right? With more variables, you do more times, then by chance you have a false positive rate. Yeah, so then you need to control that. And the second thing is, you have so many variables, you need to select which one plays the role as the target. And variable selection is very important. Okay, I think in the engineering also variable selection, like using machine learning method, is often being used, right? Yeah, and then adjust for multiple testing, like you have a false discovery rate, how to control that, yeah, FDR method. And then, so the challenge, what are the statistical or big data method we use to do that? And importantly, graphics play a very important role, visualization, right? So variable selection, false discovery, graphical method help to explore large data set. And then we do the shrinkage regularization, empirical Bayesian method, signal feature extraction, and also FDR method controlling false discovery rate. Yeah, those I think you probably already know. So I want to go to the detail. It's just give you an overview. Okay. And then the next page. So this is the graphical example how we do the first stage. So these are. So if you look at it here. So this is box plot. I think everybody know box plot. So each of these is a box plot of 40,000 genes for each particular subject. And then you can see it's going up and down. And then here I have different treatment groups, 3 different treatment groups. As I mentioned in the very beginning, I said among the 40,000 genes, the disease only impacted few of them. So in that case, you would expect from subject to subject, majority, even the median should line up, right? And then that means because we use different setting, like we have each subject being tested in a particular plate. Then the plate going through the pipetting, put the sample into different position, and then you put the treatment at each wells. So all of those can be done by different days by different analysts, technicians. So those all add to the plate effect. So what we do is we use the assumption the median should all at the same level, and then we do median subtraction. After that, you can see it's lined up together here. And then what we study are, so those are the control. So we put in each plate, we put in a control. Here it's all over the place, but after we normalize, the control roughly more straight line. And then what we study are those still being extreme, those are potential disease impacted genes. We will screen those in the second step using more accurate technology. All right. So FDR method is drug discard development is heavily regulated, and so false claim, for instance, if not working, you claim it's working, then that's a huge thing that the regulatory want to control that. So what they ask us to control that type of error by 5%. So if you do each individual gene test, it's 5%. Then you have, suppose you find 100 genes, then you add them together, it's really a much bigger false discovery. What we do is instead of using the probability of error, we use the ratio, the number of ratio to be 5%. Because you have 40,000 genes in the denominator, then in the numerator, you can have a larger, relatively larger number to still control the false discovery on the 5. Yeah, so that's the idea. Yeah. So I have a paper correspond to compare different FDR method, because different FDR method giving you different results. So in industry, we mostly interested in, you could develop a new method, but if it's a different result, which one is true, which one is not. So we did a larger and so how do we handle this? Is careful experimental design with enough power. So that's why in phase 3 study you see large sample size. Okay? Because phase 3 is the result you submit to FDA, and then you don't want to make a false claim. So the larger the sample size, and then the variability will be reduced. Once variability reduced, then you have more power. Okay? And so as you know, like if you learn statistics, it's very fundamental knowledge. Test statistics, typically in the numerator is the mean difference, and then in the denominator is the variability of the mean difference. It doesn't matter how the distribution look like. And if distribution different, then you describe the center shifting using different statistics. The variability deriving can be different formula. But in general, it's always the mean difference in the numerator, the variability of mean difference in the denominator. Imagine if you reduce the denominator, the ratio will be bigger, then you will see the efficacy is bigger. So that's why we always focusing on reduce the variability, find the source of variability. And then, so there are also methods to deal with the artifact, outlier, such using the robust regression. And the robust regression, like if you know wavelength, for instance, you use different bases, and then you find the baseline drifting, you subtract baseline drifting. And then you can also find isolated artifact using that model. And also the mixed effect model is very effective, because mixed effect model is each factor contribute to the variability can be added to the model, and then variability can be calculated for each factor, then subtract from random error. And then we also do subgroup analysis, like you can do, for instance, cluster analysis, and group the subject into different subgroups. And then each subgroup, you do the subgroup analysis and as a sensitivity instead of analyze them together. And non-parametric method is also very popular. Okay, so how to integrate analytical method with biological knowledge? The network analysis, Bayesian network analysis is being used. So this is quite advanced method being used more and more often. So just Bayesian method meaning you have to describe the structure and with technician, with the clinician to know all those variables, which one is the driver, which one is the outcome. Once you have these building blocks, then what you do is you start with a small structure, then you add in a group to see whether your likelihood increase. If increase, then you keep it in. If it's decreased, then you not put it in. So you're going through this many, many times, then those arms putting into there, you see how many times it's been occurred. So that's called the frequency analysis. So here is a real illustration. So you have so many things together, and then in the bottom is often is your outcome. Here is a clinical outcome. Here is those, for instance, in the beginning I showed you with the 13,000 outcomes. An input, you can put it here, and then you let this tree be growing, and then until you find a stable tree. You can grow this tree repeatedly using bootstrap method, like many times. And then this is like where we collect the feed, the input from the clinician. And then this is the result. And what we interested is in those clustered structure. So everything serve and point to one thing, meaning they are the cause of the disease. And then this is the center could be the T helper cell. T helper cell is very important in immune disease. Because we have T cell, we have B cell, and then if you've been infected, your T cell and B cell will fight first. So if you see certain disease, and you collect the T cell and the B cell to see whether it's caused the disease. And so now we move to lead identification. I can go quickly. So lead is the chemical compound. Over the years, the pharmaceutical company will accumulate millions of chemical compounds because chemists will synthesize those compounds. Once they synthesize, they will store there as their property. Just like in a book library, you have 1,000 of books, right? And in the chemical library, we have millions of compounds stored there. And then suppose you identify a disease target, like it's a gene, one gene, for instance. Then you take all those millions of compounds to test against that one target. So that is using the high throughput screening. It's a very lengthy experience. So for instance, here you can see you screen 700,000 molecular compound each day, okay? And then it's a high throughput screening. And then what we do is you cannot actually analyze so many data points. What you do is you cluster those many compounds into different groups by certain property. Then you just take a representative from each group to test. So that can reduce your number of testing greatly. So similarity, how do you define similarity? You work with a chemist. And then how do you run your design? It's more like data scientist expertise. Okay, so what you do is you design the experiment and you break your experiment into different elements. Like we call each run can last 3 days. And the day can be a factor, technician can be a factor, and then each factor associated with a certain variability. Yeah. And then you do the virtual screening and to see the similarity. And then you can, so approaches here, I mentioned this on like AD, toxic study, and then approaches using predictive model machine learning. As AR is a structure and activity relationship. Structure meaning the compound structure. Activity is their intensity against the target, right? And so here is a more like a formula. And so I can give you this as a more easy to understand example. So here are 2 chemical compounds. They are already on market as the effective treatment. These are their compound 3D structure. If you're a chemist, you understand this. And then we put this structure together with their compound physical measurement, like the mass, the size, and nuclear type, all those. And then you can see this will form for each compound. There's a long list. And then this will be your input. And then here, the yes or no is binary output. Binary meaning effective or not effective. And then you build a virtual screening using the machine learning method. And then, so with those two, you can serve as your center of the cluster. Then you see screen on your compound library. Then see which one more close to this, which one more close to this. Right? Then can be potential treatment for similar disease. And so now we talk about digital. So emerging technology and really generate a lot of digital data. For instance, now you can like heart rate, blood pressure, ECG, physical activity, sleep pattern is not measured at the doctor's office. It's just wear some devices in your wristband, and then you can collect continuously 24 hours a day. And then this is just showing some of the real data we received. As you can see, it's going up and down, and then some are noise, some are real peaks. And then how do we denoise and identify the pattern? So this is, bio signal process would be very useful. And the imaging data. So imaging data starting from the cell imaging. So cell has a life cycle. From healthy cell to disease cell to dead cell. So we study the morphology of the cell. And also we take the imaging of, for instance, a new treatment could be using the imaging, heart imaging, and brain imaging to study. And the challenge is like instrument. You need to validate the instrument, calibrate the instrument to measure those bio signals, right? And then you also need to do the denoise and pattern recognition, feature identification. And then once you find the feature, doing the analysis of the endpoint and develop automated analytical tools. Very important. So I did several tools for like heart rate, for EKG, and the EEG is the brain wave. And then the skill we need is knowledge and the skill related to science and technology. And the data management. And data management meaning from the device all the way to the database. And in between, you have a data management issue, you have the denoising issue, data merging coming from different sources, the scale different, you cannot treat them equally. You need to put in a weight for different sources of the data, right? So how do we develop the right weight? And then you also need to have a efficient computational and programming skills. People like as a statistician before using SAS, now using Python, you need to learn and advance your digital skills. And then you need to have the consultation, collaboration skills across different scientific functions. So each function they have called subject matter expert. So we talk to them and learn the area. And so this is the reference more related to the new technology of Díaz network if you wanted to learn, and here is the reference. So that's all of my talk. And thanks for your attention.
Two main models. One model is for description of the action of genes.
Yeah.
And this is a kind of, let's say, a static observation. And another point is the action of some molecules on the behavior of the cells, no?
Yeah. So like they measure different things. For genotype is what you can observe, right? And the clinical demographics is also the patient is. You basically can observe like a weight, the BMI index. And then there are some things not observable, like you have to use certain technology to measure like RNA sequence. proteomic cell count. And then these are the disease outcome, also the safety outcome. So with the doctor's input, we see the impact itself. For instance, one gene can impact the neighborhood gene. So we have this, we call it A can point to B, B can point to A. And then here is we call the demographic clinical. They point to here. So the direction is per discussion with the clinician and the biologist. Then these are the outcome of disease, like disease status, for instance. And then for SLE, multiple sclerosis is can impact 8 organs. Then you have really many outcomes. Then these be impacted by all of these. These will be on the left-hand side of your equation. So how to design this structure and which one impact which is you need the subject matter knowledge of the disease.
Mm-hmm. Yeah. I see. Another question? Yeah, please.
Mm-hmm. Professor, did you and your group ever think to use some data generation skills to treat the big P and small samples issue?
So we did that, like for instance, we wanted to rank certain candidates, right? So what we do is we just resampling, for instance, bootstrapping is you resample 90% of your whole population, then you put it back, resample another 90%, and then you can do it many times, 1,000 times, and then you build and stabilize the tree. Yeah, so it's, as I mentioned, this is proved by statisticians. This process will eventually converge to the truth. Good question. So I don't know if you use the same like terminology, bootstrapping.
No.
Yes. Okay. Yeah. So resampling, bootstrapping. But even you do the resampling, there are some conditions need to be satisfied. Like if you have 2 different populations, and sometimes they mix together doing the resampling, and that you need to make sure the 2 populations, their covariance structure are similar. Otherwise, it will be wrong. The result will generate. Yeah, the distribution. You can have some shift, but the covariance structure should be similar.
Yeah. Okay. Okay. Let me ask you a more general question.
Mm-hmm.
We are here in a computer science department,
Yeah.
and the students are getting more specialization in computer science.
Mm-hmm.
Would you require or would you recommend some kind of studies for the students in order to get in touch with the pharmaceutical industry?
Yeah. So I know we have a digital department. It's different from biostat department. So they build certain groups to do they call the Gen AI. So what they do, for instance, they do large language modeling. So what they do is for the past many years of question and answer, the regulator deal with the agent, with the FDA. So they build the index. So if now you receive a question, you want to see what's the answer we gave previously, and then you don't have to have a human to serve, right? So that's being already implemented. Another is using the modeling to really study the past recruitment, patient recruitment, how fast, how slow, which area we went, and then with those historical knowledge, computer scientists can say, okay, this area, likely we will recruit patients faster. So that's another. And also what we do is we do a lot of regulatory documentation, right? So instead of starting from the very beginning, we build the template, and the computer data scientist can build the template, and then according to different questions, like manufacturer question, assay testing, clinical question, and categorize them, and quickly build a template, and instead of starting from scratch. So it's also using the Gen AI.
So do you know what is Gen AI?
It's our class. Generatively.
Okay. So let's thank very much. Your very interesting
My
talk.
pleasure.
Thank you very much. I would like to give you one recognition
Wow, thank you.
redacted in Spanish and English.
Thank you so much.
Thank you very much.
Thank you.